Welcome to the November 2018 edition of AJP-Heart and Circulatory Physiology's video table of contents, or from the editor's desk. The November issue is one of the largest we've published in recent years. This is partially due to a backlog of papers that we're finally getting out, and to more and hopefully better review articles covering many areas of cardiovascular physiology. So here I'm highlighting primarily the review articles and some very few selected articles. So here's a look at what's inside, okay? In our first review this month, Dunford et al. Discuss cardiovascular aging and skeletal muscle microcirculation. They focus on the use of contrast-enhanced ultrasound, or CEUS, as a promising non-invasive technique that has been used for diagnostic medicine and more recently as a complementary research modality to investigate the response of the microcirculation in insulin resistance, diabetes, and aging. This brief report outlines the assessment of microvascular flow using the raw time intensity curve incorporated into gamma variant response modeling. CEUS could be used to complement any macrovascular assessments to capture a more complete picture of the aging vasculature, and the modified methods presented here provide a template for the general use of CEUS within a research setting. Dampney et al. Review the hypothalamic paraventricular nucleus as a unique and important brain region involved in the control of cardiovascular, neuroendocrine, and other physiological functions pertinent to homeostasis. The PVN is a major source of excitatory drive to the spinal sympathetic outflow via both direct and indirect projections. In this review, they discuss the role of the PVN in the regulation of sympathetic output in normal physiological conditions and in hypertension. In addition, they discuss the ability of exercise training to correct sympathetic hyperactivity by restoring blood-brain barrier integrity, reducing angiotensin II bioavailability, and decreasing oxidative stress and inflammation in the PVN. Boengler et al. Review the role of the mitochondria on myocardial conditioning. While several signal transduction pathways become activated by such maneuvers, they all have a common endpoint, namely the mitochondria. These organelles represent an essential target of the cardioprotective strategies, and the preservation of mitochondrial function is central for the reduction of ischemia-reperfusion injury. In this review, the authors address the role of mitochondria in the different conditioning strategies, in particular on alterations of mitochondrial function in terms of energy production, formation of reactive oxygen species, opening of the mitochondrial permeability transition pore, and mitochondrial dynamics induced by ischemia reperfusion. Ogola et al. Review sexual dimorphism in arterial stiffening, disease pathology interactions, and the influence of sex on mechanisms and pathways. Patients with increased arterial stiffening have a 48% higher risk for developing cardiovascular disease. Like other cardiovascular pathologies, arterial stiffness is sexually dimorphic. Young women have lower stiffness than age-matched men, but this sex difference reverses during normal aging. Although remodeling of large arteries is a protective adaptation to higher wall stress, arterial stiffening increases afterload to the left ventricle and transmits higher pulsatil pressure to smaller arteries and target organs. The authors conclude that additional studies may be needed to elucidate the mechanisms by which females are protected from arterial stiffness. Kin et al. Tested the hypothesis that chronic supplementation of exogenous humanin, HN, a protein with cytoprotective effects in middle-aged mice, could prevent and reverse cardiac fibrosis and apoptosis in the aging heart. HN treatment significantly increased the ratio of cardiomyocytes to fibroblasts in aging hearts, as shown by this percentage of each cell type in randomly chosen fields after immunofluorescence staining. Furthermore, the increased collagen deposition in aged hearts was significantly reduced after HN treatment, as indicated by picrocereus red staining. HN treatment also reduced in aging mice cardiofibroblast proliferation and attenuated transforming growth factor beta 1 fibroblast growth factor 2 and metallomatrix protease expression. These results of the present study suggest a role for the mitochondrial-derived peptide HN in the cardioprotection associated with aging. Kawamoto and colleagues aim to elucidate the effect of aging on shear mediated dilation of the common and internal carotid arteries. Hypercapnia-induced shear mediated dilation in the common and internal carotid arteries were assessed in 10 young, 5 men and 5 women, and 10 older, healthy adults. Shear mediated dilation was induced by 2 levels of hypercapnia and was calculated as the percent rise in peak diameter from baseline. There were no differences in shear-mediated dilation between younger and older adults in either artery under low levels of hypercapnia. However, shear-mediated dilation in response to higher levels of hypercapnia was attenuated in older compared to young adults in the internal carotid artery, but not in the common carotid artery. These data indicate that aging attenuates shear-mediated dilation of the internal carotid artery in response to high levels of hypercapnia, and shear rate is an important stimulus for hypercapnic vasodilation of the internal carotid artery in both young and older adults. Callahan and Greevy measured wall shear stress made on the aorta of 224 subjects with normal anatomy using 4-dimensional flow MRI using multiple encoding velocities and an optimized post-processing routine. The spatial and temporal variation in wall shear stress and oscillatory shear index were analyzed using a flat map representation of the unfolded aorta. The influence of aortic shape and velocity on wall shear stress were evaluated using regression analysis. Wall shear stress in the thoracic aorta is dominated by axial flow with a strong negative correlation with advancing age. The spatial distribution of wall shear stress is highly heterogeneous, with a localized region of elevated wall stress along the length of the anterior wall seen across all individuals. These data demonstrate that the accurate 4-dimensional flow-derived wall shear stress measurement is feasible and can provide a standardized parametric approach for presentation and analysis. Thank you for listening, and once again, please go to our AJP-Heart and Circ webpage to take advantage of all the great content that you won't necessarily get on PubMed. It's been a pleasure putting this issue together. We will see you again in December. In the meantime, have a great Thanksgiving in the US and Canada, and don't forget to send us your best work now.